Hi, welcome back to Introduction to Bioengineering. We talk about cancer this week because it's a leading cause of death in the United States. As people age, the likelihood that they contract and die from cancer increases. On this graph, you can see age of people in the US and the cause of death for people of that age. And what I hope is clear is that cancer becomes more prevalent as you get older and older. I think this graph is really interesting to look at, and I encourage you to come back to it to sort of think about, how old are you and what are the things that are most likely to bring about your demise if you were to pass away soon. I want to point out that data is from 2017 and that there have been changes in cause of death across age groups in the US in recent years. Those are mostly due to the COVID-19 pandemic and rise in opioid abuse in the US. And so some of this will have shifted around a little bit, but the kind of growing incidence of cancer with age and the high fraction of people that it kills, this is around 30% of people 55 and older, is still very significant. Okay. Cancer is a relatively new phenomenon. If you look at causes of death in Boston in 1812, so about 200 years ago, you'll see that 5 out of about 1,000 people have cancer recorded as their cause of death. A lot more people are dying from tuberculosis or diarrhea, all kinds of other maladies, fevers of different types. And one of the reasons why people are dying of cancer is because of what was shown on this previous slide. You have to live to be a certain age in order to have your likelihood of cancer increase significantly. So that's part of it. People's life expectancies were not as long 200 years ago. They may also not have recognized certain things as being cancer. And so you may have passed away because you got a fever, but the underlying condition that was leading you to be susceptible to a new type of infection, for example, could have been a cancer. So an increase in our ability to diagnose cancer and then a lengthening in lifespans has brought about more and more cases of cancer in time. Now, what is cancer? Simply put, cancer is a disease in which some of the body's cells grow uncontrollably and other parts of the body. On this slide, you're seeing a dividing breast cancer cell all the way on the left-hand side. You can see the DNA here in blue and the tubulin, which is sort of organizing DNA during this process of division. The green is marking mitochondria, so the kind of energy powerhouses of the cell. And you can see just that it's more or less a normal cell, but it's dividing. And then on the right, you're seeing mammograms, which are essentially X-ray imaging of a patient who has very early stage breast cancer that's indicated by this little box. And what you'll notice is that it's more or less normal, but it's the cells in this area that have started to divide when they shouldn't. Now, I think an easy question to ask point is, aren't cells supposed to grow, right? Why is it dangerous that cells in your body are dividing? But although cells in your body are supposed to grow, they usually grow or multiply in order to fulfill a need. For example, they may replace or old cells, but when they grow and multiply when they should, then they start to form tumors or little lumps of tissue that can either be cancerous or not cancerous. And whether you're cancerous or not is really just a measure of how fast you're growing, how sort of dangerous you are to the rest of the body. Now, cancerous tumors can spread into nearby tissues and can travel into distant parts of the body to form new tumors. This traveling into distant parts of the body is a process called metastasis. And cancers become lethal when they interfere with the normal functions of organs and cells in your body. So you can actually live for a pretty long time with a tumor as long as it's not interfering with the function of the organ that it's growing in. So for example, if lung cancer, in this image, you're seeing stage 4 lung cancer. So this is pretty advanced. The tumor is this sort of whitish region you can see inside the lung. If it were not interfering with lung function at all, if you were able to breathe normally, even with this big mass of cells growing inside of your lungs, and actually wouldn't be that dangerous. The problem is that these cancer cells often interfere with the function of the organ. So in this case, interfere with your ability to breathe, which is lethal, right? And if some of those cells break up from your lungs and travel via your blood system to other portions of your body, then you can get cancers or tumors forming in new locations that may be even less amenable to the growth of new cells. There are more than 100 types of cancer. Cancers are usually named for the organs or tissues where they're from. So this could be lung cancer, brain cancer, etc. They may also be described by the type of cell that formed them. So they could be epithelial cells, squamous cells. Sometimes you'll hear a combination of both the tissue that they're from and the cell type they're formed. And if you want to, you can go to this link at the bottom of the page and scroll through all of the different types of cancers that we currently categorize or kind of break cancers up into. So you can read about each of them, and I encourage you to do so. It's kind of astounding how many different types of cancer there are and how differently they behave. There are two sort of broad categories that we're going to break down cancer into in this lecture. There are solid tumors. So this is a solid mass of cancer cells. The breast cancer tumor that you saw on a previous slide, that's an example of a solid tumor. But you can also have liquid tumors. These occur in blood, bone marrow, or lymph nodes. And these are usually the product of a cell that is normally found in blood or lymph fluid growing uncontrollably. So in this case, on the slide, you're seeing an example of leukemia, where these white blood cells are replicating and taking over the blood. Essentially, you're getting too many of them, and that is what is liquid tumor. And it's different solid tumor in that it is in circulation kind of throughout your body all the time. Here's just an example of a biopsy, blood biopsy, and you can actually see the sort of cancerous patient's blood sample has a few of these white blood cells in it, and then the cancerous or person who has leukemia sample has a lot more of them. And they're just colored dark in this case because of a stain that's applied to the white blood cells or the blood in total before it's imaged on a microscope to make the blood cells easier to see. They don't actually look that dark most of the time. Okay, so if those are the types of cancer we're going to talk about, solid tumors and liquid tumors, then I think a good question is what causes cancer, right? We know that it's an overproliferation of cells, but cancer, like what causes them to overproliferate, right? Cancer is caused by changes in DNA, often in gene coding regions, but not always. So sometimes you get a gene that gets mutated, and that makes the gene either non-functional or hyperfunctional. And we'll talk about what that means in a second. So mutations that turn on oncogenes or inactivate tumor suppressors are the most dangerous. These are ones that are most likely to cause cancer. Now, these are two different jargon words that I just threw at you, and so let's define them. Oncogenes are genes that promote uncontrolled cell division and growth when they're constitutively activated. So these are genes that stimulate your cells to grow and divide. Usually, these genes are very tightly controlled. They're only expressed when you need to make a new cell to fill a function within the body, for example, to replace a dead or dying cell. But when they're turned on constitutively, which means all the time, then you can get cells that they constantly need to be growing and therefore start to create little tumors. So mutation in an oncogene is one that will activate it. HER2 and KRAS are just examples of genes that are commonly found to be turned on. These are oncogenes in cancers. Tumor suppressors, on the other hand, are genes that would usually slow or stop cell growth. These are inactivated in cancers. So in a normal cell, a tumor suppressor gene is intact, And in a cancerous cell, you may have damage to your DNA that deletes or somehow inactivates one of these tumor suppressors. And without the tumor suppressor, without the expression of something like P50 to stop cell growth, you start to get uncontrolled cell growth and formation of a tumor. Okay. So mutations to oncogenes or tumor suppressors are what can lead to cancers. Here's an example of a very famous tumor suppressor. So you may or may not have heard of BRCA1. BRCA1 is a DNA repair protein that's expressed in breast and other tissues. The protein structure is shown on the slide right here. So this is a ribbon diagram showing you sort of the alpha helices or DNA binding regions of the protein and some business end of it. The protein is involved in error-free repair of DNA double-strand breaks. So your DNA gets broken all the time. Different things that you encounter in the environment can cause damage to your DNA. UV light is a good example of something that can cause damage to your DNA. This BRCA1 protein will come in and fix that damage, and it fix it without errors, which is really important because that means you're not getting mutations in genes within your genome wherever this BRCA1 protein is expressed. Now, BRCA1 is serving this DNA repair function in breast tissue and in ovarian tissue and pancreatic tissue to a smaller degree. And when it's mutated, then you get more DNA damage in these tissues. So if you have a mutation in BRCA1, which occurs commonly actually in the human population, this is something that can be passed from generation to generation, from mother to child, or from father to child. And when you hit a damaged BRCA1 copy, then your risk of getting breast cancer in women goes up 50 to 65%. Or risk of getting pancreatic cancer is around 1 to 3%. Your risk of getting ovarian cancer is 40 to 65%. That's greater than a coin toss for both breast cancer and ovarian cancer. So it's pretty severe. It's really significant that people have figured out that this one gene is so important for cancer risk. Now, the discovery of BRCA1 has led to some really interesting behavior, some interesting prophylactic measures. So this just means sort of preemptive measures to try to prevent the patient from getting cancer. In this case, what you're seeing is actually Angelina Jolie, a rather famous actress who elected to have a double mastectomy. So she had both of her breasts cut off because she discovered that she actually carried a broken copy of BRCA1, which meant that her risk of getting cancer was incredibly high. And so she chose to remove the breast tissue to remove the chance she might one day get breast cancer because she had this lack of a functional BRCA1 gene. And actually her decision to do this, she was very vocal about. She published an opinion piece in the New York Times and talked about it on a number of daytime talk shows, and actually led to a number of other women getting screened for BRCA1 mutations. They wanted to know, do I carry also a dysfunctional copy of the protein? And for women who found a mutation in their genome in this BRCA1 gene, they were also electing mastectomies and hysterectomies, which is the removal of their ovaries. And she spurred, or this sort of increase in prophylactic mastectomies is called Angelina effect, actually, as people sort of tell their doctors, oh, I heard this actress talking about her decision to have a mastectomy because I want to know if I have this same mutation. So sort of interesting that this is one approach to avoiding, albeit a rather extreme one, but is one that people are pursuing. How BRCA1 is got some interesting history beyond just Angelina Jolie. A biotech company called Myriad Genetics first located BRCA1 gene on the human genome or human chromosome number 17 in the late 1990s. They actually established the first clinical lab to commercialize genetic testing. So they've established first genetic test that you could get prescribed by your doctor, and they saved countless lives by informing women about their BRCA1 risk. But they also patent the BRCA1 gene sequence and cancer promoting mutations. So they patent the sequence of the fully functional gene and of versions of the gene that would activate it that are commonly found in people. And this patent prevents other companies from running BRCA1/2 diagnostic tests from looking for this same gene. So it's great for Myriad Genetics that they have essentially a monopoly on this type of test. It's not so good for patients because the cost of getting one of these tests becomes really high. Myriad Genetics can essentially charge whatever they want. They have competition. And patients can't get second opinion. So if Myriad Genetics comes back and says, no, you're fine, you don't have this BRCA1 mutation, but a patient sees so much familial history of breast cancer or ovarian cancer and they want a second opinion, there's no place to get them. And so this actually causes a legal battle. And genetics is sued by the Association for Molecular Pathology. And this course case goes all the way to the Supreme Court. And this court rules actually against the molecular Myriad Genetics, excuse me. They decide that you cannot patent a DNA segment, which is really fascinating. Actually, it has some interesting ramifications on the current kind of state of diagnostics in the country and of biotech. Here's a quote from the majority opinion, which was written by Chief Justice Clarence Thomas in 2013. He said, a naturally occurring DNA segment is a product of nature and not patent eligible merely because it has been isolated. However, cDNA is patentable because it is not naturally occurring. So that's interesting. They basically decide you cannot patent BRCA1 or its naturally occurring mutant versions. And so that opens up other companies to be able to make BRCA1 genetic tests and sort of do the same thing that Myriad Genetics has been doing. But they leave the door open for patenting cDNAs. So cDNA is complementary DNA. It's DNA that's made in the lab in vitro from RNA. So you start with an RNA, a message, and you build DNA that is essentially the complement of that RNA. And those sequences you can patent. So while this did throw Myriad Genetics' business model into a bit of turmoil and actually make it really difficult for the company to continue to develop new genetic tests and profit from them, it didn't completely scare off innovation in biotech or in diagnostics altogether, as people worked to figure out how to kind of avoid running foul of the Supreme Court and work with cDNA in order to get patentable genetic material. Here's just a picture of the Supreme Court in 2013, if you're curious about the people who made the decision about cDNA and whether or not humans can patent naturally occurring DNA. Okay. So if genetic changes can underlie cancer, then a good question is what causes genetic changes? You can inherit specific genetic sequences from your parents. That's sort of how the BRCA1 mutation is passed from generation to generation. UV light, though, and chemicals, smoking, these things in your environment can also cause damage to your DNA. Viruses, and then even just the process of cell division can also cause damage to your DNA. And because each of these exposures increase with age, you get more and more DNA mutations as you age. And that is part of why your cancer risk goes up as you age. So here's just a cartoon example showing a child who has had very limited exposure to harsh chemicals, to viruses, has had less cell division because he just hasn't grown that much. And as this child ages, they may encounter more and more DNA damaging agents in their environment and start to accumulate some mutations. And that process just accelerates as the person ages. And so your risk of cancer later in life is greater because you have had so much more exposure to things that may be damaging your DNA. Now, how can we detect cancer is a really big question in the field, because we know that early detection typically results in higher survival rates. Here are statistics for non-small cell lung cancer, for stomach cancer, and pancreatic cancer, showing that if you catch the cancer in stage 1 or 2, your chances of survival are much higher than if you catch them at stage 3 or 4. Literally half to maybe even a third or a quarter of the survival rate if you catch it later than if you catch it earlier. So it's really advantageous to figure out whether or not you have cancer as early as possible. Now, it brings us to a whole section that we're going to have here on how cancer is diagnosed. So no single test diagnoses cancer. It's because there are so many different types of cancer. So it's really challenging to have one test that can tell you whether or not you have cancer at all. It may be able to tell you whether you have breast cancer or not, but it probably can't simultaneously detect a lymphoma or another type of cancer. Okay, so cancers are found via symptom screening test. You may show up to the doctor and say, I'm not feeling very well, and the doctor rules out a bunch of, very specific or acute causes of whatever pain you're feeling. Say you have stomach pain. They may, test you for different types of pathogens to see if you picked up food poisoning somewhere, look for problems with your kidneys or appendix. Maybe you have kidney stones. But they can rule all of that out. They might start to run tests that look specifically for cancer. Another way that you may get cancer testing done is just by age-related screening. So for women after the age of 40, you start to get mammograms done, which are those types of X-ray tests we showed earlier, looking for breast cancer or cancerous cells within your breast tissue. There's a similar type of screening that's done for men for prostate cancer. For people of both ages, they get screened for colon cancer via colonoscopies after, I think, the age of 55. But unless you're falling into one of these categories where you either have symptoms or you're of a certain age that you would require screening, it's unlikely that you're going to get a lot of cancer screening early in life. Now, what do these screens look like, or what do these tests look like? You can get a blood chemistry test. This looks at sugars and salts, different components of your blood, just to see how well your kidneys, liver, things that should be filtering the blood are working. And if you see, very strange results from a blood chemistry test, that may tell you like, oh, the kidneys are not working that well. Maybe we should do a follow-up test of some kind to see if there's something wrong with the kidneys. This may be an imaging-based test, and we'll talk about what those are in just a minute. Another pretty straightforward and routine type of test would be a complete blood cell count. This just looks for red and white blood cells. It counts basically how many of each type you have. They can be used to diagnose things like leukemias. Remember the slide I showed you earlier where there were these microscopy images showing you lots and lots of white blood cells in a person's blood sample? That would be a way of diagnosing a leukemia, basically saying there are more white blood cells here than we would expect. You may also have more white blood cells if you have an active infection and those cells are trying to fight off that infection. And so you do sort of have to decouple some of the blood cell count information from other things that may be making you sick, right? Like a high white blood cell count may not just be because of cancer. So you sort of have to use these things with all of the knowledge you have about a patient. Okay, you can do cytogenic analysis. You can basically take a sample and look at people's DNA and see if there are changes in chromosomes. Are there any broken, missing, or rearranged? This could tell you that cancer is likely because these changes in chromosomes where pieces are obviously broken missing or arranged are so severe that it could very well lead to cancer. You could get immunophenotyping. This is where you take the tissue and you try to stain it with different antibodies. And those antibodies will identify specific proteins, typically on the surface of cells. And you can use the kind of protein content on the surface of a cell to diagnose and stage or monitor blood cancers. So certain blood cancers will have a high concentration of very specific surface markers. And so you can know what type of cancer you have by what type of antigens stick to the surface of your blood cells. You can do what's called sputum cytology. And so this is just, sputum is sort of like what you would cough up from your lungs. And if you analyze that, you can look at sort of what cells are getting coughed up from your lungs. If you have cancer, you may get an unusually high number of cells and of cells, just acted sort of cellular mass in your sputum. So that's one way of searching for, in this case, a lung cancer. You could look for tumor markers. So these could be proteins or other substances that are typically made in higher amounts by cancer cells than normal cells. So if you see a high number of tumor markers in a blood test, then you would assume or it is possible that you would have cancer cells somewhere in your body producing all of these proteins and substances. You can analyze urine. This is a way of diagnosing kidney, blood, urethral cancers. And if any of these sort of bodily fluids tests come back positive, you may follow it up with some based tests. So you can use CT scans. These are essentially a series of x-rays that create 3D images in the body. You can look for things like colorectal cancer or lung cancer this way. Often you'll ingest contrast agents that sort of help with these scans to make it clearer where within your body cancer cells are active. You can get MRIs, which use radio waves to image the inside of a body. This can also be used to find solid tumors. You can use PET scans. This is another one where you would ingest a contrast agent before getting a PET scan. And it shows you where glucose is taken up in the body. And if you're ingesting or if you're using a lot of glucose in one location that's expected, then it might be that you have cancer cells there, because cancer cells will consume a lot of sugar, a lot of glucose in order to grow quickly. That's another way of searching for cancer in the body. And then you may get a biopsy where a little bit of tissue is actually cut out of a tumor and it is analyzed under a microscope to see kind of hallmarks of disordered growth. And you can actually stain tissue that you collect from a biopsy to look for cell surface markers using antibodies, sort of in the same way that we were talking about these immunophenotyping tests before. These biopsies can help people stage and diagnose different types of cancers, not just blood cancers, because you're actually taking samples from solid tumors in this case. Okay, so here's all of these different tests, and they're all good. They complement each other in different ways to be able to identify lots of different types of cancers. But can we do better than that? That's something that bioengineers have been asking for a while and trying to figure out an answer to. Because if we can get better early detection of cancer, we can increase the odds that people survive. So what about a better blood test? Here's an example that I really like, in part because it's pushed forward by a professor here at Stanford. This is work out of Steve Quake's group. And here they're using cell-free DNA to try to detect pancreatic cancer at one of its very earliest stages. Now, what is cell-free DNA? It turns out it is exactly what it sounds like. It's actually DNA floating around in your blood that is not within a cell. How does it get there? Well, if tumor, then there may be a lot of unhealthy cells, and unhealthy cells because they're trying to grow so fast, maybe lysing and releasing DNA from their insides, just kind of letting it out. And that DNA can very easily enter the bloodstream and end up circulating. And so you can collect a blood sample and actually look at just the DNA that is not housed within cells. You can, by centrifugation, separate whole cells from just DNA. And then you can sequence that free DNA and you can see, is there anything unusual that I'm seeing in this free DNA? Because you will get some free DNA coming from healthy cells as well. What they found is that there's a lot of cytosine signatures on non-cancer cells that are missing from pancreatic cells. And so if you do sequencing of cell-free DNA and you look specifically for this type of modification and you don't see very much of it, then it's likely that you actually have a pancreatic cancer developing within the body, which is pretty amazing because pancreatic cancer is actually one of these cancers that is most frequently detected very late. It does not cause a lot of symptoms. And by the time patients are losing weight and feeling ill, they often have very aggressive pancreatic cancer with typically months left to live. And so something where you could diagnose pancreatic cancer from a blood test would be really powerful because it could mean that you could get routine screening for this type of cancer, well before you have any type of symptom. Okay, so actually this type of screening is being done commercially, not necessarily for pancreatic cancer in this case, because this 5-methylcytosine modification is a newly identified signature for pancreatic cancer cells, but there are other cell-free DNA markers that you can look for. And this company is selling a test that will allow you to search for 50 different cancers with a blood test. Here's just a workflow for how this type of thing works. We have patients that they collect blood samples from. In the clinical trials where they were trying to establish that this type of screening test worked, they would randomize essentially these samples, and some of them would as a control that are just stored for future testing, but other ones are run through their kind of proprietary test to see whether or not the patient's blood contained DNA that would indicate that they had a specific type of cancer. And then they would refer patients who had a positive test, who might have cancer, to further diagnosis and assess to see whether or not their tests were correct. And they were good enough to get approval to be sold. These tests run about $1,000 per test. So for $1,000, you can get tested for 3 different cancers all at once. And the idea is that you would do this on healthy patients to screen for cancers that they don't even know that they have, for things that are not really causing any symptoms. So I think that's really powerful and can be very impactful if we can really screen people regularly for cancers in an invasive way. That would be great. Is $1,000 at $1,000 a test? How frequently do you want to do this? I'm not sure. It's actually something I would love for you guys to ponder and consider. Okay, what else can you do for early cancer detection? I showed you this image already, and what I think is really fascinating about this is actually in this early image, these are the same patient taken at different time points, and you can see that there's actually almost nothing within this red box, right? I would not certainly have looked at that and said that looks like a cancerous mass to me, especially as it looks pretty similar to other parts of breast tissue. Later, after the breast cancer develops, you can more clearly see that there is a tumor growing there. The innovation here is actually from a professor kind of MIT's lab. She's using image based deep learning models, so machine learning models, to predict breast cancer up to 5 years in advance. So it can take these images, they've been trained, this model has been trained on a sea of breast cancer images at different stages, and the machine learning model can now pick out areas that might look to us like they're totally normal, but that are likely to develop into cancer in the future. It can help women get earlier diagnosis for breast cancer, or just know that there may be problematic areas to follow up on and potentially get screening more regularly than they have otherwise. So, a pretty cool use of machine learning for cancer diagnosis. Okay, cancer treatments. How do we actually treat cancer? How can we do this better than we're currently doing it? Here's an example from another Stanford bioengineering professor's lab. This is Professor Jennifer Cochran. They recently engineered a novel cytokine ligand in order to increase or rather slow down cancer cell growth in mice. And so what they did was engineer this cytokine CLCF1 in order to have tighter affinity for one of its binding partners. You can see sort of the kind of complex assay that they set up in order to do that is cartooned right here, where the cytokine ligand would be bound by different proteins. Usually I use fluorescently tagged antibodies to detect whether or not the ligand was bound, and you can run these flow cytometry plots, try to see whether mutations that you make to this protein increase binding affinity or not. You can read a lot of details about it in this paper if you're curious. I guess the thing I would like to show you is just that the engineered version of this CLCF1, when administered to mice, actually helped keep the tumors that mice had from growing. And so they were able to have a much lower tumor volume relative to mice that were not treated with this engineered peptide. So here's a really nice of trying to slow down growth that works really well in mice. Will be interesting to see as they move it to people whether or not this new cancer treatment, this protein-based cancer treatment, would be effective. Okay, so probably the biggest thing in cancer treatment right now is something called CAR T therapy. CAR T stands for chimeric antigen receptor T cell therapy. This type of therapy actually weaponizes your own immune system to fight cancer for you. It's quite clever and a little bit complicated. I'm going to do my best to walk you through this. So in CAR T therapy, a patient gets a blood draw, and they remove T cells, a certain type of immune cell, from the person's body, and then they engineer these T cells to have chimeric antigen receptors. So T cells are usually the cells that kind of sniff out and destroy invading cells within the body. And they do so because they have little antigen receptors on their surface, and those tell the T cell when they those bind to things, and when they bind to things, they tell the T cell, hey, I found something that shouldn't be here, you should destroy it. And so the CAR T is an engineered T cell that has had a new engineered into it. You actually get genes encoding this new receptor inserted into the T cell genome. And those new receptors recognize cancer cells. They recognize proteins that are prolific or abundant on cancer cells. Now, this isn't easy to do because, again, cancer cells are sort of the same as your own cell in the body. They just have gotten a little out of hand. They're replicating too much. They're dividing too much. So they have a lot of the same surface markers, a lot of the same proteins on their surface as healthy cells. It's just they have a higher abundance of certain types of markers on their surface, certain proteins on their surface, than healthy cells do. And so CAR T therapy takes advantage of this and says, well, maybe these T cells will kill some healthy cells in the process, but they will mostly kill cancerous cells. So the chimeric antigen receptors, these sort of cancer recognizing proteins that are put onto the surface of the T cell, have been very specifically picked out in order to help maximize killing of cancer cells and minimize killing of healthy cells. Once the T cell is engineered, it is what's called expanded in vitro. So it's just stimulated to grow itself so that you get millions of these T cells. And then they're infused back into the patient who we originally took the T cell from. So your own cells in the process are getting taken out of your body, engineered, and then put back. And when they're put back into your body, they should find cancerous cells within your body, use the chimeric antigen receptors to bind to them and to start to kill them. And incredibly, this stuff works. This is an article celebrating the 10-year anniversary of a child's first CAR T therapy. There was a 6-year-old woman named Emily who had an acute lymphoblastic leukemia, and she was treated with CAR T therapy and has survived. She's now 16 and still cancer-free thanks to this type of therapy, which is pretty amazing because she had had many unsuccessful rounds of chemotherapy, and her disease was progressing so rapidly that she was ineligible for bone marrow transplants and other things that are often used to treat leukemias. And this was really a last line of defense for her. It was a brand new technology at the time, and it is pretty amazing that it has worked so well. Since Emily, lots and lots of people have received CAR T therapy, and it's been great. It actually helped cure cancer in lots of people worldwide, but it's without controversy. So CAR T therapy is incredibly expensive. It costs anywhere from $500,000 to around $1 million per sort of patient that gets treated. It's expensive because of the types of manipulations and treatment involved. So for each patient, you need to collect T cells, you need to run biopsies to sort of see how their cancers are progressing. You need to actually engineer those T cells so that they have antigen receptors being expressed from them. You need to reintroduce these CAR T cells into the patient, and they are monitored after this infusion quite a while, and so they stay in the hospital for a while. They get imaging studies and medicines done kind of all the time in order to ensure that this CAR T therapy is going well. There's actually, the stuff is that WebMD actually has a whole page dedicated to navigating the financial aspects of CAR T therapy. You can actually read that down here. I thought that was interesting. There's also, when this was first coming out, a lot of stories in the news about how expensive this type of therapy actually is. But what we pay for cure for cancer, right? It's sort of a difficult thing to put a dollar amount on. I think most people would pay a lot to save their only child or their young child if they had the means to be able to do so. But who really has the means to pay $1 million for a therapy, right? So just an interesting aspect of RT. There are many insurance companies that do cover this type of thing now, but it is an active area of debate to figure out how to bring cost of therapies that have been expensive to develop down in price, and to make them actually impactful for people, because if they are too expensive, it really won't have the reach that it could. Okay, so in addition, I want to emphasize how incredible I think CAR T therapies are, but then also give you full sides of this, right? I think it's an important part of being well versed in bioengineering is to really not just think about how good certain things are, but also like what are the challenges that we're still facing with some of these new technologies. And with CAR T therapy, there's still a lot for us to learn. We're about 13 years now after the first CAR T treatments. And since then, researchers have found instances of cancer relapse. It's only about 1/5 to 1/3 of patients that go into complete remission. So they never ever have a relapse, which is still pretty good, better than a lot of cancer therapies, but not perfect. We have identified patients for whom the therapy just does not work. And as far as I know, they still don't really understand what it is that leads this type of therapy to work really well for some patients and less well for others. They have found instances where CD8 cells, so this is the type of T cell that's usually being engineered to express these chimeric antigen receptors, turn into CD4 cells, which is a different type of T cell. CD4 cells are more like helper cells that sort of assist in the killing of things, whereas CD8 cells are called killer T cells because they're the ones that are actively destroying invaders with the body. So sort of a mystery to what it is that's causing CD8 cells to turn into CD4 cells after the CAR T therapies eliminated cancer from a patient's body. Is it just that that's how immune systems sort of naturally progress? What is going on there? It's sort of an interesting area of research for people to dive into and continue to explore. In some patients, there are serious side effects for CAR T therapy. You can get a high fever, some have slipped into comas, get really low blood pressure. Typically, these are acute and resolved pretty quickly, but there have been a couple of instances where patients have not survived administration of CAR T therapy, and that is still not very well understood. And is another thing to figure out for the type of treatment. It's rare, but it does happen. Okay, and then CAR T therapies are not successful in solid tumors. Okay, so we'll get back to the solid tumors in a second. Now, CAR T therapy has been under kind of constant technological innovation. People are constantly trying to make this stuff better, and there have been some really incredible things that have been done to try to improve CAR T therapies. I'm just going to talk about two of them today, but I encourage you to dig into the literature to find out more interest in CAR T therapy. So here's an example where researchers have improved CAR T therapy specificity. So I mentioned that that tumor cells are not that different from healthy cells because they are essentially derived from your healthy cells. So they are you, they're just sort of your cells out of control. And they may have more copies of specific proteins on their surface than a normal cell, but it's the same protein that's on the surface. And so if a CAR T cell recognizes this specific surface protein, then it may go about killing a bunch of normal cells in addition to killing tumor cells if it has a linear relationship with antigen recognition and killing. So that's sort of shown here. This engineered CAR T cell would kill both this normal cell and the T cell. And so Rogelio Hernandez Lopez, who is also a professor of bioengineering at Stanford, developed an approach to change sort of the sensitivity of the CAR T cell to the amount of antigen displayed on the surface of a human cell. And so the way this is supposed to work is that at kind of low concentrations of surface markers on these cells, you would get no CAR T cell killing, and only if there was a high concentration of the surface marker, which would indicate that this is actually a tumor cell, would you get which is essentially killing of that cell. Okay, how does this actually work? Here's a little schematic for how this type of CAR T cell kind of ultra-sensitive switch works. I'm not actually going to go through it in this lecture, but it's here if you want to think about how this might work. There's a low affinity switch, a high affinity switch, and one kind of leads to the other. And that's the way that they engineered this ultra-sensitivity. Some data I want to show you where they tried putting two different types of tumors into mice. One is a very slow-growing non-HER2 tumor. And so HER2 is an example of one of these antigens that may be on the surface of a normal cell at a low concentration and on the surface of a tumor cell cancerous cell at a high concentration. And when mice who have these different types of tumors on their flanks are injected with CAR T cells with their ultrasensitive switch, they are able to completely stop tumor growth of the tumor that has a high concentration of HER2 and really not attenuate the growth of the other cells all that much. It's just trying to show you that this type of CAR T cell is able to very specifically pick out cell types in the body based on the amount of surface protein that they're showing. This is kind of an extreme example where both of the cell types they're monitoring are in fact human tumors put into mice, but it is showing you that the kind of circuit that they enter into these T cells is actually working. The T cell is now able to pick out based on the concentration of HER2 on the surface of the cell, whether or not it should kill the cell or not. Okay, so that's Rogelio's work. I highly recommend you check this out. The other thing I want to talk about are solid tumors, because solid tumors, as I mentioned, are difficult to treat with CAR T therapy. So in contrast to blood and lymph cancers, things that are called liquid tumors, which move through blood vessels, it is difficult to access the cancerous cells in a solid tumor. So you can imagine if this mixture of cells is floating around in blood and mixed with a bunch of T cells, the T cells could easily act all of these extra lymph cells and destroy them, all of these cancerous cells. But if the cancer cells are within the solid mass of tumors or of a tumor, then it can be difficult for the T cells to get in there and start killing off the cancer cells. And that's a problem because many different cancers come from solid tumors, right? Here's a PET scan showing you a lung cancer. Here's a brain cancer, colon cancer, liver cancer. All of these are very different types of cancer, and they're all solid tumors and not currently treatable with CAR T therapy. Okay, so why is it? Well, it turns out that typically the tumor microenvironment, sort of the conditions in a solid tumor, suppress T cell activation. That's what this little red bar is trying to show you. And that includes cytokine production, which can help sort of stimulate T cell activation. And so the bioengineers have bypassed the suppression by engineering synthetic IL-2 circuits into T cells that are activated independently. And so the tumor, therefore, is not, how do I explain this? The surface marker that would usually be recognized by the T cell would be the triggering event to activate the T cell. But because the tumor microenvironment is sort of suppressing this inactivation and preventing this IL-2 production, which feeds back and sort of helps reinforce activation of this T cell, you don't get T cell activation in the case. Here they've sort of decoupled the two. And so now when the T cell recognizes surface marker that it's interested in, it produces its own interleukin-2, and that kind of helps reinforce T cell activation. And this T cell kind of ignores the signals that the tumor is sending to stop its activity. And what you can see in these images on the right are that CAR T cells would normally get kind of stuck, sort of black dots along the top at the surface of a solid tumor, which is here. Whereas when they have their engineered CAR T cells with this interleukin-2 circuit, they you good injection of the CAR T cells into the solid tumor, and you can see them already sort of destroying this tumor environment. Also exciting. So this is work done by Professor Kyle Daniels. He's a new professor at Stanford and is continuing to push forward CAR T therapy in exciting ways. Okay, so those are the two kind of CAR T advances I wanted to highlight for you guys today. And, you know, where is this going next? Well, there's so many different types of new CAR T therapy modifications being developed and tested to improve this technology to make it better for solid tumors, to make it more specific and have fewer side effects. And so, I would encourage you to check this out. I wanted to put up this picture of a naked mole rat because I think they're cute and they're fascinating because these rodents actually don't get cancer. And so there's something that people have been interested in studying, have been studying for quite a while to figure out, like, why is it that these rodents don't cancer? And can we steal any tricks from them? People have figured out relatively recently is that one of the reasons why mole rats don't get cancer probably has to do with a special hyaluron in their extracellular matrix. This kind of sugar is something that they think helps with the elasticity of the naked mole rat skin. But when they interfere with production of this compound in the naked mole rat, they actually do start to get tumors forming. And so it's thought that maybe the molecule, or really the kind of structure of the extracellular matrix, is important for tumor or cancer cell development and progression. And so there are questions about whether this molecule could help humans avoid cancer. There's a lot of active research being done with naked mole rats. In addition to CAR T cells and new methods of diagnosing cancer and new, protein-based treatments, all kinds of stuff, there really is a ton of work being done on cancer and all these different cancer types. And so I encourage you to dig deep and see, what interests you in cancer and in bioengineering specifically. Okay, here are reflective questions for today. How often would you want to be screened for cancer? Let's say that you had access to these tests, $1,000 per test for 50 plus cancer. Is that something reasonable? Would you do it every year, 5 years, every 10? If you only did it every 10 years, is that still effective? I just need to think about the current state of cancer screening and cancer diagnosis, and what you wish to change about it, or what you think about it now. I think it's kind of incredible that we can engineer our own immune cells to fight cancer. But what other diseases could we fight with this capacity to engineer T cells, to reprogram them to find something that they wouldn't usually find in your body and just it? Where would you look in the naked mole rat to learn the secrets of its cancer-free life? I don't really know the answer to this, but I thought it would be fun for you to contemplate it. I only skimmed the basic CAR T engineering here, so I want to encourage you now to go learn more about it. You can read links to some of the news articles we had here if you want to know more about Emily, that young girl who had CAR T therapy when she was 6, or if you want to know more about the kind of costs of CAR T therapy. If you want to know more about the technologies, you can read the links to the scientific papers that are there. Whatever you found to be most interesting, I highly encourage you to dig deep on it and to learn more. All right, thanks so much.